Hello everyone, welcome to my channel. Today in this video I am going to discuss about the topic is mitochondria. So mitochondria, what is mitochondria? Mitochondria is a cell organelle which is present in all the eukaryotic cells. So mitochondria is a double membrane bound organelle. See this structure, it is a double membrane structure. This is the inner membrane and the outer membrane. So this organelle is double membrane structure. If we say singular, it is mitochondrion. Mitochondrion means one. Singular mitochondria. Mitochondrion. Mitochondria is plural, means more than one mitochondria is present in a cell. Mitochondria are double membrane bound organelles of eukaryotic cells. Occurrence will be they are found in eukaryotic cells except mammalian RBCs. Mammalian RBC what? In the RBC, this mitochondria are absent. Why mitochondria is absent in RBC? Because these RBCs, what does it do? It takes oxygen and what is the function of RBC? The function of RBC is to transport oxygen to all parts of the body, or it gives oxygen to all the cells. So if RBC will have mitochondria, then it will consume oxygen. That's why RBC, the matured RBC, lacks these mitochondria. And in RBC, there is an anaerobic respiration will takes place in RBC. And these mitochondria are absent in prokaryotic cells. There are two types of cells, that is eukaryotic and prokaryotic. In the eukaryotic cells, mitochondria is present, and in prokaryotic, mitochondria are absent. These are the cell organelles. Now shape. The shape of the mitochondria will be oval, sausage shaped, spherical. These are the shape of the mitochondria. You can see the structure, it looks like a sausage shape, or you can say oval shape. The measurement or length of the mitochondria is 1 to 5 micrometer. Mitochondria is having two membrane, that is the outer membrane and the inner membrane. This is the outer membrane and the inner membrane. Mitochondria have two membranes protecting it on the outside. This outer membrane will help in protecting it from outside. The outermost layer smooth and also contains transport proteins that passes material in and out of the mitochondria. Second, the inner membrane folds inward. This inner membrane, can you see, it is folded inward, and these are called cristae. This infolds are called cristae. What does it do? It increases the surface area for the synthesis of energy. This is called cristae. And they are the site for ATP. And this is adenosine triphosphate. These infolds allow more surface area for ATP synthesis. Next is origin. Mitochondria have a small and circular DNA and 70s ribosomes of their own and make some of their proteins. So in this, there will be circular DNA is present in this. Circular DNA will be present in the mitochondria and 70s ribosomes of their own. The mitochondria will have DNA and 70s ribosome. What is 70s? 70s is sedimentation coefficient, and these mitochondria helps in the replication of and the mitochondria will have their ribosomes, and these ribosomes helps in the formation of mitochondria. The membrane of the mitochondria, it helps in the, these ribosomes helps in the formation of membrane, mitochondrial membrane. These ribosomes of mitochondria, what does it do? It helps in the formation of membrane of mitochondria. Now next is structure. Mitochondria are semi-autonomous. What do you mean by semi-autonomous? They are mitochondria and the plastids are double membrane, and they have their own DNA. These organelles which have their own DNA, they can reproduce independently of the nucleus. Without the help of nucleus, they can replicate by itself. That is called semi-autonomous. The inner membrane is selectively permeable to small ions and molecules. Mitochondria are semi-autonomous organelles bound by an envelope of two unit membranes and filled with matrix. In this, there will be a matrix will be present, that is inside the mitochondria. The outer membrane is smooth and has porous protein which forms channels for the passage of molecules through it. The inner membrane is selectively permeable. So the inner membrane, that is called cristae, is selectively permeable to small ions and molecules. It will be selectively permeable for ions and molecules. This cristae increases the surface area of the mitochondria to hold a variety of enzymes, and this increases the surface area for the production of ATP, and this matrix will contain various enzymes for the synthesis of ATP. Now next is, what is the function of mitochondria? Mitochondria are the main site of cellular respiration in eukaryotic cell. It oxidizes different food products. Now what is cellular respiration? What is cellular respiration? Cellular respiration means the glucose oxidizes. Glucose oxidize. What does it mean, oxidize? Breakdown of glucose. Breakdowns into pyruvic acid. It produces energy in the form of ATP. Storing calcium. So what is the another function of mitochondria? It helps in storage of calcium. So the calcium is quickly absorbed in the form of calcium ion in the mitochondria. And calcium is required for muscles, and it is important for blood clotting. These are the functions of calcium. Next is, what is the function of mitochondria? Cell death. Cell death is an essential part of and regeneration of new cell. As cell damages or becomes old, they are destroyed by the mitochondria. So these are the functions of mitochondria. Next is Golgi apparatus. This Golgi apparatus is another cell organelle which is discovered by Camillo Golgi in the year 1898, and it is single membrane structure, single membrane covering. In plants, Golgi body is known as dictyosomes. Where it is located? It is located near the nucleus. So it is located near the nucleus, and its structure is pleomorphic structure. What is pleomorphic structure? Means its components of Golgi body are different in structure and shape in different cells. In different cells they will have different structure. Or it pleomorphic means it has the ability to change their shape and size. Change shape and size according to the environment. Golgi body is always directed towards the cell membrane. This concave side is directed. This is directed towards, if this is a cell membrane, so the concave side will be directed towards the cell membrane, that is called transphase, and the convex surface towards the nucleus. So here I'll show you the structure. See this side, this is the convex side and facing towards the nucleus, and it is called cis phase. This is called cis phase, facing towards the nucleus, and concave side facing towards the cell membrane. So this is the cell membrane, facing towards the cell membrane is called transphase. Cis phase. Golgi body is always directed towards the cell membrane and is called maturation phase or transphase, while its concave surface is directed towards nucleus and is called formative phase or cis phase. Structure will be, this Golgi apparatus will have 4 types of structural components. It has 4 structural components. First one is cisternae, tubules, vacuoles and vesicles. In this structure we will see, this is the Golgi apparatus. The cisternae is arranged in a stacks. They are arranged in stacks. So these are the cisternae, and these are vacuoles. These are vacuoles. These are vesicles, and these are tubules. These are tubules. These are vesicles, and these are vacuoles, and these are cisternae. There are the 4 components of Golgi apparatus. Cisternae, they are long, flattened and unbranched saccules. 4 to 8 saccules are arranged in a stacks. So these are arranged in a stacks. Tubules, these are branched and irregular tube-like structure associated with cisternae. They are associated with the cisternae. Vacuoles. So vacuoles, these are the vacuoles. These are vacuoles. Large spherical structure associated with the tubules. Vesicles, these are the vesicles. Spherical structure arise by budding from tubules. Vesicles are filled with secretory materials. So some secretory materials will be present in this. Secretory materials will be present in this vesicles. Now what is the function of these Golgi apparatus? So what it helps in the secretion and acts as storage, modification and condensation or packaging of products within the vesicles. So this is the vesicles. In this, the materials which are coming into the Golgi apparatus, what does it do? It helps in storing. It is storing the material, modifying the materials, and what does it do? It packages. Why it is packaging the material? To transport the materials into the cell or out of the cell where it is required. Second point is, it forms acrosomes of sperm. So sperm, what does it do? So if this is a sperm, this is a part called acrosome. The specialized vesicle present in the head of the sperm filled with enzymes which dissolves the egg cells. It helps in dissolving the membrane of the egg cell at the time of fertilization. Next point is it forms lysosomes and secretory vesicles. So it helps in the formation of lysosome. Lysosome is another cell organelle which is present in the cell, and the function of lysosome means it contains some digestive enzymes which helps in, or it is also called suicidal bag. At the time of cellular damage, these lysosomes burst and release enzymes and they digest its cell. Next is it helps in the synthesis of cell wall material. It means it helps in the synthesis of polysaccharide. Polysaccharide means it helps in forming the things which is required for formation of cell wall. Formation of glycolipids and glycoproteins. What is glycolipids and glycoproteins? These are helps in the formation of cell membrane. Formation of cell plate during cell division, dividing the two cell. So these are the functions of Golgi apparatus. In next class, we will discuss about other cell organelles. Thank you.